Okay, so here are our second messenger systems. We are going to do 3. These are just the 3 most common ones. There are variations of these, many different variations of these, but these are just kind of the general 3 main examples. So here's one, and here's one. So there are 3 of them that we're going to go over. These 2 are very similar, so these 2 are easy. You have your receptor. You have the receptor hooked to a G protein, and that G protein is inside the cell and it's inactive. Then along comes the neurotransmitter. I'll just abbreviate that as NT, that for neurotransmitter, and it binds to the receptor, and that activates the G protein. And once the G protein is activated, it in turn is going to activate an enzyme called adenylyl cyclase. What adenylyl cyclase does is it converts ATP into CAMP. CAMP is a second messenger. This is an enzyme itself, and this is going to stimulate the cell. Now, depending on what kind of cell this is, it can do that by activating enzymes or kinases. We don't need to get into the difference between those 2 right now. They just act very similar. They're both going to be stimulatory sorts of things within the cell in this case. Or it can open ion channels too. So like a sodium channel, for example, or a calcium channel. It really depends on what this cell is. It'll have various different effects depending on what that cell is. So that is our, that is our second messenger system there for CAMP. Now the negative feedback for this, to stop CAMP, so once you've had the effect of CAMP, you want to stop it, is an enzyme called phosphodiesterase. And phosphodiesterase is found in the cells all the time. And once CAMP does what it needs to do, then the phosphodiesterase can break it down so it doesn't just continue to stimulate that cell. The next second messenger system that we have, very similar. We have our receptor with the G protein inactivated inside the cell, bound to it. The neurotransmitter binds to the receptor, it activates the G protein, and the G protein activates an enzyme. This time the enzyme is called guanylyl cyclase, and what guanylyl cyclase does is it takes GTP that's found within the cell and it converts it into cGMP. And cGMP is going to stimulate the cell in the same way that CAMP did, whether by opening ion channels or activating enzymes, kinases. It's going to stimulate the cell. So these 2 really very similar pathways here. Even the negative feedback is the same. It's phosphodiesterase again. Phosphodiesterase is going to break down cGMP. And we're going to see this in our next unit, Unit 8, when we do vision, because cGMP is really involved with the visual pathway, and phosphodiesterase breaking it down is important for you to be able to see anything. So we're going to see this one again. Okay, now for our third second messenger system, on the surface it looks a little more complicated, but it is really just the same idea of just a little series of chemical reactions. It's just that there are 2 happening at the same time. So let's just walk through it. It won't take much longer than these other 2. It's really not that much more complicated. Because we start out the same. Receptor with a G protein inactive, bound on the inside of the cell membrane. Here comes the neurotransmitter. The neurotransmitter binds to the receptor, activates the G protein, and that G protein activates an enzyme. And that's just like what we saw before. This enzyme now is called phospholipase C. What phospholipase C does is it goes to the cell membrane and it kind of snips out little pieces of those membrane phospholipids. It's not going to break a hole in the cell membrane or anything like that. It's just using those phospholipids on the inner cell membrane to activate 2 other enzymes. Those 2 enzymes are inositol triphosphate, or IP3, and diacylglycerol, or DAG. They're both being activated at the same time, or they're both being formed at the same time. So they're going to be acting simultaneously, but let's do IP3 first. So what IP3 is going to do is it's going to go to the endoplasmic reticulum, And on the endoplasmic reticulum, you'll find IP3 receptors, which is where IP3 is going to bind. And once the IP3 receptor binds there, it opens something called ryanodine channels on the endoplasmic reticulum. When those ryanodine channels open, calcium is able to come out of the endoplasmic reticulum where it's been sitting there in storage. So we get calcium coming out into the cytoplasm. That was the whole point of IP3, was to get calcium out of storage and put it out here to where it can interact with diacylglycerol. So once the calcium interacts with diacylglycerol together, they can activate the last thing, protein kinase C, or PKC, and that stimulates the cell. So really not quite as complicated as it looks at first glance, right? Okay, so the last thing that we need to do for this unit then is to, I'll try to leave this here because we're going to refer to it back and forth. We're going to look at two different neurotransmitters and the different kinds of receptors that we can have there. And these are very basic examples of the receptor types that you have for these neurotransmitters. For example, like for this alpha-1 receptor, there's lots of little subtypes under there. Same thing for all of them, really. So we're not going to go into that sort of detail. We're just looking at the neurotransmitter and for each one, main types of receptor. So let's start with epinephrine and norepinephrine, what we would collectively call adrenaline. And we all know what adrenaline is. It's the sympathetic nervous system. It's activating, fight or flight. It's stimulatory, that sort of thing. So the receptors that epinephrine or norepinephrine can bind to are called adrenergic receptors. So if we go back to our picture here, let's say this was epinephrine or norepinephrine, then this receptor has to be an adrenergic receptor. One of these. Because if it's a different kind of receptor, it's not going to bind the epinephrine or norepinephrine. So our four different kinds of adrenergic receptors. We have alpha-1, alpha-2, beta-1, beta-2 receptors. For the alpha-1 receptors, those are G-protein coupled. All of these are G-protein coupled. The alpha-1s are going to use phospholipase C as a second messenger. So, for example, this could be an alpha-1 adrenergic receptor. This could be epinephrine or norepinephrine. And then all of that is going to happen when epinephrine or norepinephrine binds to it. Okay, now an alpha-2 receptor involves phosphodiesterase. So let's say that we have another receptor on the cell that, say, is over here. And it activates phosphodiesterase. That could be an alpha-2 receptor. Now what does phosphodiesterase do? It breaks down CAMP. It breaks down cGMP. Both of those things are stimulating a cell. So if we have more phosphodiesterase inside of a cell, we're going to be inhibiting a cell. We're going to be slowing the cell down. With phospholipase C, this one, for example, we're stimulating a cell. With phosphodiesterase, we're inhibiting a cell. And you might think, wait, adrenaline is stimulatory, but it doesn't stimulate everything. It stimulates, say, your skeletal muscle. It stimulates your heart. It stimulates, blood flow to your brain, but not so much blood flow to your digestive organs. You're inhibiting your digestive organs, things like that. So you do need to inhibit some things for sympathetic effects. So that's where this sort of receptor comes along. Okay, for the beta-1 adrenergic receptors, those use CAMP for a second messenger. So this could be a beta-1 adrenergic receptor. Epinephrine or norepinephrine binds to it. All of this happens. We produce CAMP as our second messenger, stimulate the cell. That could be a beta-1. And then beta-2, those activate phosphodiesterase. So they do that. So they inhibit. So this alpha-1 stimulates a beta-1, stimulates a cell, where